Good afternoon, everyone. Welcome. I'm Jack Van Horn from the University of Virginia, and it is my distinct pleasure to welcome you to this edition of our 2024-2025 Foundations of Biomedical Data Science seminar series. Our unique lecture series is made possible through the generous support of the Integrated Translational Health Institute of Virginia, the UVA College of Arts and Sciences, the UVA School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since its founding in 2016, the Foundations of Biomedical Data Science seminar series has been at the forefront of exploring the transformative impact of data science methodologies, artificial intelligence, deep learning, and statistical modeling across diverse themes in the biomedical and health sciences. Our selected theme for this year is focused on quantitative approaches in spatial multi-omics for guiding personalized medicine, where each Friday we enjoy presentations from a wide range of leading thinkers about the issues, the promise, opportunities, and hurdles associated with advanced genomics, transcriptomic, metabolomic, and other omic methods, along with a wide variety of data analytic opportunities presented for data science, deep learning, AI, and spatial modeling approaches. Participants selected for our Biomedical Data Science Innovation Lab program utilize the content from these lectures as essential material in their culminating in-person manuscript and grant project development workshop, which this year will be held at the Hutton Hotel in beautiful downtown Nashville, Tennessee in June of 2025. We are very excited for this year's program where junior faculty, biomedical, and computational experts come together to develop joint projects on quantitative approaches and applications for spatial multi-omics and their data types and their use towards guiding personalized medicine approaches. For today's seminar, I am really delighted to welcome our speaker, Dr. Jill Barnholtz-Sloan from the National Cancer Institute. She is an acting director of NCI's Center for Biomedical Informatics and Information Technology, or CBIIT, and she is the associate director for Informatics and Data Science program within CBIIT, and she leads efforts to shape data science, data sharing, technology strategies with the ultimate goal of fostering collaboration within NCI and across the NIH and the cancer research community. In addition, she maintains a robust research agenda in descriptive epidemiology and etiology of brain tumors as an intramural NCI researcher in NCI's Division of Cancer Epidemiology and Genetics Trans-Divisional Research Program. In the spirit of our 2024-2025 Biomedical Data Science Seminar Series theme, her lecture today is entitled Utilizing Data for Advancing Cancer Discoveries, where she will remind us that data is everywhere and data sharing is now required by many funding agencies as well as periodicals, and that large-scale multi-omic data sets have been utilized and continue to be utilized to make significant advancements for all human health, including cancer. And that the Cancer Institute is providing access to data resources from a number of large NCI-funded studies, which in addition also include cloud computing workspaces and analytical workflows, which are also available. As always, we are streaming this lecture live and for recording via YouTube. So if you're watching us on YouTube, wherever you are in the world, thank you for joining us. Also, our specially selected 2024-2025 Biomedical Data Science Innovation Lab participants, as well as any past alumni, are encouraged to participate by submitting any questions they have via the chat feature in their YouTube sessions. I will be synthesizing these questions and I'll ask them on your behalf during the last 10 minutes or so of the lecture period. With that, thank you so much, Jill. We are really looking forward to your presentation.
Thank you so much. I want to thank Dr. Van Horn and the entire UVA team for having me here today. I'm really excited to be here to talk with you all about data just being everywhere and data being an enabler. I will be using cancer as the use case as I talk to you about various things that are available. But you really could think about utilizing data for enabling discoveries in any human disease. I do not have any conflicts of interest, but I am a full-time paid employee of the National Institutes of Health. So let's talk just very quickly about the importance and goals of data sharing. So data sharing is the practice of making research data and metadata available for use by the broader community. And the NIH expects all funded research to have a plan to manage and share their scientific data that's generated and to make their publications broadly available. And this is really about promoting open science, stimulating new discovery, so data being that enabler for new discovery. Also about rigor and reproducibility and transparency. And it's really about a cultural shift, thinking about just collaborating within your own group or a collaborative group or consortium that you work with, to broader data sharing and how to have the most impact from the data that you are using for your own questions, but could possibly be used to answer other questions. At the NCI, we have a spectrum of data access that goes everywhere from fully unrestricted, fully available, no constraints, to a controlled access process. And the informed consent is the basis for institutions to determine the appropriateness of submitting data on this spectrum of data access. And hopefully some of you have used these different layers of access to get access to some of our data assets. So I want to talk to you today specifically about some big data assets that we have available at the NCI, those in the Cancer Research Data Commons or CRDC, and those also available in the Childhood Cancer Data Initiative or CCDI. So let's start with the Cancer Research Data Commons. It is meant to empower all of you by providing a secure, accessible cancer data ecosystem. Within the ecosystem, we provide cloud computing resources, visualization tools, analytical tools, so that you can interoperate in a flexible, cloud-based computational environment that is secure and also with sort of in your domain of influence to create your own secure workplace for your team. And so we are very interested in moving forward with this infrastructure, which I will tell you more specifically about, but making sure that we are lowering the barriers so that anybody in the cancer research community can access and use the data available. Here are just a high-level view of some of the statistics and impact. Data available in the Cancer Research Data Commons is on over 130 unique subjects. And we have over 10 petabytes of data. And you can see that we have lots of users and lots of different workflows and compute power available. We recently published, thank you to Cancer Research, which is one of the American Association for Cancer Research journals, for hosting 4 papers for us. 2024 was the 10th anniversary of the Cancer Research Data Commons, and you can learn more specifics about what I'm talking about today for CRDC by taking a look at those 4 papers. So let's talk specifically about what types of data are available. Within our CRDC ecosystem, we have multiple different data commons. They are typically focused on the type of data that's available. We do have a new data commons coming very soon, the population science data commons, which will make some of the large-scale epidemiologic data assets available. The genomic data commons is the oldest data commons. It's been around the longest. And that's where the Cancer Genome Atlas data is, which is a very highly used data set from the NCI data assets. We also have the proteomic data commons, which includes all types of omic data in the proteo and proteogenomic space. We have the imaging data commons, which shares medical imaging, MRI, PET scans, CAT scans, and uses the DICOM standard to share those. We have the integrated canine data commons, which is a very interesting data commons that allows you to cross-compare canine data and human data. There is a little bit more of a focus on immuno-oncology for those trials in the canines in there. The Cancer Data Service has been our broad cancer repository that takes any type of data. And so there are lots of different data assets available there. And then we have a new one, the Clinical and Translational Data Commons. It currently includes data from the Cancer Biobank, which was a moonshot project, but it will soon have clinical trial data available in it. We have lots of different tools that allow you to get access to these data so that you do not have to download them yourself. Let's talk a little bit about our cloud resources. There are 3. And they each have different things that they bring to the table that make it easier to interface with the data sets. And really what it allows you to do is you get funds to get you started so that you don't have to pay the cloud costs up front. We help you with that. And you can bring your own data and tools to then leverage the data assets that we have available there and all of the analysis tools and workflows and pipelines to analyze your data. So hopefully you will take a look and there'll be something interesting there for you to use. In addition, we now have a centralized data submission to the Cancer Research Data Commons, which we hope helps investigators with their data sharing expectations. And so we accept new submissions. You go through this submission request form, and then you will get contacted, and then you can actually submit your data through here as well. And all of this in the Cancer Research Data Commons, there's lots of interoperability that has to occur between the data commons. And we do that through a data standards service. So we have semantic harmonization and a shared data model across the Cancer Research Data Commons. And then we also have a cancer data aggregator, which is an API layer that will allow you to query and aggregate data across all of Cancer Research Data Commons. So just to give you an idea of what the data standards service does is it links information on, this is just giving you an example on sex at birth, subject reported ethnicity, anatomic site, and disease type, and how we take information that could come in multiple different ways, and we make sure that it fits with our cancer dictionary public definition, and also our NCI thesaurus concept code. So these are just a few examples of how we do that. So when you submit data, we help to map it to this common data model. The Cancer Data Aggregator allows you to search across all of the CRDC data sources using harmonized common language terms that, of course, are built off of the data standards service and the metadata for all the data that's available. You can use it through an API interface, and you can use a simple query language to then allow you to generate a listing of all the individual patients and all the data sets that are available, all the different types of data that are available for them. And then you can either download those data as long as you have permission to access those data, or you can use them within one of our cloud resources. Let's shift gears a little bit and talk about the Childhood Cancer Data Initiative. The goal is to gather data from every child, adolescent, and young adult diagnosed with cancer from around the country, so that we can learn from every child and improve the quality, consistency, and accessibility of data so that we can develop new knowledge and better treatments. There are a couple of things that the CCDI is doing that's really exciting. They are establishing a federated pediatric cancer data system. You can see in the graphic here on your right, all the different types of data that they are trying to link together so that they can have more data available for discovery and potential novel clinical discoveries. And these are all the different, many of the different data sources that are coming into the Childhood Cancer Data Initiative. And I'll tell you a little bit about the ecosystem that they have available now that is available to all of you to go and explore and use for analysis and access to the data. As you can see, it's a really big, broad range of data and ways that you can interface with these data, which is really great. In particular, I encourage you to look at the CCDI Hub, where you can find not only data sets, but also tools that will help you analyze these data. And you can actually interface with the Hub through an Explorer dashboard, which is very point and clicky. And it provides you with a faceted search and visualization of search results so that you can export results for further analysis. Let's shift gears a little bit. I'd love to tell you about two very specific examples on how these data assets that I just talked about, specifically the data assets that are available in the Cancer Research Data Commons, have driven change, really, really important change impacting cancer. So the first is we talked about the Cancer Genome Atlas. These data are available in the Genomic Data Commons. The Cancer Genome Atlas has been around for a while. You can see this really exciting and interesting arc of papers that they have published. These papers have been transformational for the different tumor types that they study. And there's over 11,000 patients and almost 3 petabytes of data available. And it includes all types of genomic, proteomic, imaging, and clinical data. I myself am a brain tumor researcher, so I want to tell you a story using the Cancer Genome Atlas data and the impact that it's had for brain tumors. And I was just honored to be part of these Cancer Genome Atlas teams looking at both glioblastoma and lower grade glioma. So when you look at the broad spectrum of brain tumors, there are malignant brain tumors and non-malignant brain tumors. Malignant brain tumors are quite rare. The most common type of malignant brain tumor is a glioma. And so that's what the TCGA focused on. The most common type of glioma is a glioblastoma. Brain tumors are not staged. They are given a histologic type. Now they're looking for hallmark molecular features, and they're given a grade. So a glioblastoma is a grade 4 tumor. And I think here is where this just really exciting impact of big data comes into play that led to changes in the WHO classification for how brain tumors are diagnosed. So on your left, you see a Kaplan-Meier curve, a survival curve from the glioblastoma paper from 2013. And you can see that there's really something very different about the purple hash line that's at the top that seems to have a greatly improved survival from everybody else. It turns out that that hash line includes individuals with glioblastoma who have an IDH mutation. And then leading from that, for the lower grade glioma study, you can see here that the schematic is now very definitive that in order to actually diagnose specific types of gliomas, we need to know the IDH mutation status, and we also need to know the 1p 19q chromosomal deletion status. And in fact, that led to changes in both the WHO classifications from 2016 and the WHO classifications for 2021. And just in the green boxes here, you can see now that instead of saying diffuse astrocytoma or glioblastoma, it has in there some of these hallmark molecular features. And these changes were driven by those TCGA studies. And then these data on these important hallmark molecular features started to be collected across the United States in the cancer registries and through the Central Brain Tumor Registry of the United States, which is a partnership with the National Program of Cancer Registries at the CDC and the Surveillance, Epidemiology, and End Results Program at the NCI. The Central Brain Tumor Registry of the United States publishes now at the population level for the entire United States what is the proportion of individuals who have these hallmark molecular features by which grade of tumor they have. So this is just showing you IDH mutation status. You can see on the left, if you just focus on the yellow component, that the grade 2 tumors have a much higher proportion of IDH mutation than the grade 4 tumors. It's very low in the grade 4 tumors. One of the other molecular subtypes that's collected is for a common type of childhood brain tumor called a medulloblastoma. It's an embryonal-based tumor, a neurodevelopment-based tumor. And there are 4 very important subclassifications that you can see here. And so you can just see that this is the distribution by age group in terms of which subtype. And there is a lot of unknown information still here, but as time goes on and these important hallmark molecular features are collected over time, the unknown percentage continues to go down. And also, why are these important to collect? Because the overall survival varies greatly depending on which group you're in. So if you just focus on the black solid line, those are the IDH wild type glioblastoma. That's the most common type of glioblastoma. That's about 95% of all glioblastomas fit in that category. But if you look at the solid dark gray line, which is in the middle of the survival curve, those are the IDH mutant grade 4. They're now called astrocytomas, but those would have been called glioblastomas in the past. Those are the folks that were in that hashed purple line in that Kaplan-Meier curve I showed you. So it's really been changed how we diagnose glioma forever, which is very exciting. I want to also give you an exciting example using the proteomic data commons data, which a lot of that data is generated from the Clinical Proteomics Tumor Analysis Consortium, or CPTAC, to decipher the pan-cancer immune landscape. So for those of you that are interested in multimodal analytics and trying to understand the tumor microenvironment across cancers and how that relates to the immune system, this paper was published by Dr. Deng's group in 2024. You can scan the QR code if you want to look. But what they did was they looked at the data across the Cancer Research Data Commons, focusing a lot on sort of this multimodal analytics, genomic, epigenetic, transcriptomic, and proteomic data. They took information from over 1,000 tumor samples and 10 cancers to try to classify tumors into immune subtypes and correlate those with clinical outcomes. They did a lot of really advanced analytics, and some of the key findings were they had 7 distinct immune subtypes. They were able to correlate those with progression-free survival and tumor microenvironment immune cell load. And they were able to identify specific kinases that were activated in certain subtypes. And so it allowed them to have this multidimensional view of tumor biology. And they developed some interactive web portals that allow you to look at these data and further interrogate them. And because of these specific kinases that they identified, they're moving forward with some very specific therapeutic strategies for some of these groups, which is really exciting. Since the group is very focused this year on spatial transcriptomics and spatial analyses, I thought I would highlight for you some of the things that we are doing at the NCI in the AI and cancer space. Really excited to tell you about a collaboration with ARPA-H, where we really want to try to unlock, connect, and translate biomedical data to catalyze health solutions. And so our project is called the Biomedical Data Fabric Toolbox. And it's about trying to solve the problem of data is just generated in large volumes and with a lot of diversity just all the time, all over the place. And it can be very hard to access, integrate, and also keep secure at the same time. And because of this, it maybe is preventing folks to get access and ask their important and interesting questions so that novel discoveries could come and help to cure disease and save lives. So the Biomedical Data Fabric Toolbox project is about trying to break down these silos and revolutionize how health data is managed and used. So we want to make biomedical research data easier to use. We want to reduce manual effort for data integration and harmonization. We want to develop new capabilities and tools. And we also want to build health data science models that can be applied across disciplines. So the Biomedical Data Fabric Toolbox involves 5 technical areas. So technical area 1 is research data capture. How do we automate some of that data collection and also extract it and make sure that the data are de-identified and therefore secure for folks to access and use. In technical area 2, it's about curation, harmonization, quality control, quality assurance of the data. How do we do that easier than we're doing it now? We still do a lot of that manually. So it's really about data, metadata, provenance, standardization. And then technical area 2 is about user workspaces and data exploration tools. And technical area 4, which as you can see is at the bottom of the slide here and runs across all the other technical areas, is really about user testing, right? Making sure that the ways that we're doing things now, if the ultimate goal is to make data as accessible and usable as possible, are we doing it correctly so that anybody in the research community could access it? And then technical area 5, which is a little out of scope right now for the Biomedical Data Fabric Toolbox, but still being worked on by ARPA-H, is about how do we just take some of these big assets and make sure that they're interoperable and generalizable. So I'll tell you a little bit about each of the technical areas. So for technical area 1, the main objective is to develop automated capabilities for secure near real-time collection of high-fidelity biomedical research data from lab and clinical settings. And so there are 4 performers who have been funded under technical area 1. You can see them here, and I'm not going to read a lot of the details, but just tell you that it runs the gamut of federated learning programs to be able to access data, automatic data capture, so like an AI-assisted human-machine interface, near real-time capture of electronic health data, again, using AI and natural language processing to look at symptoms and outcomes, and also make sure that we have the full trajectory available of clinical data. And then how do we make sure that we're capturing all the information that's available in the electronic health record, not just the structured data, but all of the unstructured data that's in clinical notes, pathology notes, radiology notes, and leveraging some large language models to do that. Technical area 2 is about developing open source capabilities that allow automated data model mapping, transformation, and curation of data. And you'll see that there are 4 folks who are funded under this technical area. And it's all about using tools, many AI-driven tools, to help curate the data so that we can get away from all of the manual curation that a lot of us have been doing. And there's a lot of leveraging of large language models and other sorts of AI-driven approaches to do this. Technical area 3 is about designing, developing, testing, and implementing intuitive AI-powered dashboards with really easy to use, very intuitive data exploration tools and data visualization techniques that are going to support the full gamut of individuals who are in the health research community. And I think that this is really, really exciting. You can see the 4 performers who are funded under this. Again, leveraging multiple different approaches and working together so that we can make sure that the data are truly accessible to anybody, no matter what their training or title of their job. I think right now, a lot of the data assets that we have available, you have to be pretty quantitatively inclined and skilled in order to really get the most out of them. So we are really excited about this collaboration with ARPA-H and all of the performers. And so stay tuned on some of that, but I'm happy to answer any questions. I also want to tell you a little bit about, this is a Moonshot funded project, a collaboration through the NCI and the Department of Energy that was really about leveraging exascale computing to try to really make a difference in cancer. And so there are 3 projects that were funded under this collaboration. The collaboration will be closing this year. And I want to tell you a little bit about the 3 projects. Admiral, which is AI-driven multi-scale investigation of the RAS-RAF activation cycle. Improve, which is innovative methodologies and new data for predictive oncology model evaluation. And Mosaic, which is modeling outcomes using surveillance data and scalable AI for cancer. So I'm going to tell you a little bit about each of these. So for Admiral, it's just really interesting. They developed this novel multi-scale molecular dynamic modeling framework that they call MUMI to specifically look at the signaling pathway of the KRAS oncogene. And they were able to use this modeling framework to identify molecular interactions between proteins and lipids that are very important for this particular oncogene signaling in KRAS. And by using these exascale computing that's available at the Department of Energy, it allowed them to really develop a detailed understanding of the lipid-protein interaction at the membrane surface that revealed states that could be exploited for drug discovery in this really critical pathway. KRAS is a very important oncogene in many cancers where it is turned on. And so they are now also deploying this computational capability to model additional membrane-associated complexes. They've also been testing out the MUMI framework in just a regular cloud-based environment, and they're getting some really interesting results there and positive results there, so that they would be able to make the framework available for anyone to use, and you wouldn't have to have access to exascale computing. For Mosaic, they developed a deep learning natural language processing algorithm that is used on pathology reports that are submitted electronically from each state's cancer registry to the NCI SEER program. And why is this important? It's really important because a lot of this had been done in the past by manual curation by a cancer registrar. And so they've been able to fine-tune these models that allow them to look specifically for hallmark biomarkers, for recurrent status, for metastasis status, and also initial diagnosis. And they've developed these computational methods in a way that they can be extended to other elements of the electronic health record and other clinical studies. And it's just really exciting. They have about 98% accuracy of their algorithms on these electronic pathology reports. And it's made a big difference for how quickly the data can come in to the cancer registries on newly diagnosed cancer cases. For Improve, they've developed and curated multiple computational models to predict drug responses. They've curated over 30 community-developed data-driven models, and they've released the initial model comparison framework on GitHub. The link is there for you. And they have groups that are helping to also generate novel data that they are going to make available. So they've looked at and compared the data-driven models that are available in the public literature on public data, and they're now generating additional data sets that will go into their data repository that will be, it will all be made available. If you are interested in looking at some of these computational resources that were made available, models, data sets, software, use cases, publications, through the NCI DOE collaboration, I really encourage you to go to computational.cancer.gov. And as these next 6 months go by, as we're wrapping up these projects, there'll be even more available there for you. In addition, in CBIIT, in the Clinical Translational Research Informatics branch, led by Umit Topologu, they're doing a lot of different things utilizing large language models for specific and directed tasks, hoping that they can really have an impact in the clinical space. And I want to tell you specifically about something that they're doing with informed consent. So what are the critical elements of informed consent? Simplification and readability, layperson descriptions, being able to generate a first draft, language translation, right? So not just in English, but other languages available for patients and their families. And then making sure that the required elements that are supposed to be in informed consent are actually there. And so it's really about making sure the right information is there, that it's at a level of comprehension that anybody would be able to understand, and that folks understand that, you know, it's voluntary, right, for them to sign it. And so we were interested, the team was interested in looking at Can they actually take informed consents that have already been approved by the Institutional Review Board and are in use? Can they use the large language models to actually translate to Spanish? Given that we have over 68 million people speaking a language other than English in their home, where more than a third of those are speaking, are reporting that their English is maybe not at the same level as the other language that they've grown up speaking. And so if we look at the cost for human translation, they can, there's a very wide range there. And the turnaround time takes quite a while, right? An average of 10 days, but it can take much longer. If we look at the possibility of how the large language models can help us with machine translation, it is much, much cheaper and can be almost instantaneous to a few minutes. So the preliminary results looked at overall the proportion of satisfactory sentences were very similar between the human translation and the large language model translation. And they were looking at specifically 7 quality metrics, comparing human to the large language model: spelling, punctuation, preservation, representativeness, flow, correctness, and accuracy. And you can see that AI had very slightly higher acceptability in 5 of these metrics. So we're very excited about this, and we've been working closely with the NCI Clinical Center on helping them to get a bunch of their consent forms translated for some of their ongoing clinical trials. In addition, the NCI, through the Office of Cancer Centers, has been supporting 6 Federated Learning Network members or sites, all through the NCI Designated Cancer Center framework. You can see that we have 2 different frameworks here. We have one that includes the NCI, University of Pennsylvania, and Oregon Health. And the second includes Hawaii, Moffitt Cancer Center in Tampa, and Wake Forest in North Carolina. They were all asked to come to the table where they could include information on these basic model cards and very specific technical specifications that they had to have available for their compute at their institutions where their data sets, and also asking for the minimum data set to be very specific on the number of patients available and specifically what variables they had available. And I think it's very exciting. So they're really looking at how can they deploy a multimodal analytical framework through the federated learning networks. And as you can see, each of the different institutions has put forward different ideas that they would like to build the models and then federate the models across the network. And it's a really wide range of ideas. So it's very exciting. And they are in the process of starting to deploy some of these multimodal models, and they are having some very good preliminary results. In addition, Daoud Mirzaman, who oversees the Cancer Genomics and Bioinformatics branch in the Informatics and Data Science program at the NCI and CBIIT, has developed a fully automated pipeline for federated learning. This is one of the models that they are going to be deploying through the federated learning network that I just described to you, where they're taking a multimodal approach and looking at WSI whole slide imaging on the cancer patients, and then also the transcriptomic data that's available, and then building a multimodal model from the information that they're able to glean from the whole slide imaging with the paired transcriptomic data that was generated on those tumors. And they have already been deploying this multimodal model across the federated learning with really, really interesting, exciting results where the model seems to be running pretty well, but they are still fully evaluating that. So, you know, data science is a team sport, definitely when you're studying something as difficult as cancer. And I want to thank Jeff and Marcos from the CBIIT leadership team all of our federal staff and contractors. If you're interested in learning more about CBIIT, I encourage you to go to datascience.cancer.gov. I want to also thank the Office of Data Sharing, who oversees the Childhood Cancer Data Initiative, all of their staff and contractors. My own research team in the Division of Cancer Epidemiology and Genetics, some of the information that I shared with you from the Central Brain Tumor Registry of the United States and some of the work we did with TCGA with brain tumors. I wouldn't be anywhere without my research team. If you're interested in learning more about the Division of Cancer Epidemiology and Genetics, I encourage you to go to dceg.cancer.gov. If you're interested in brain tumors, which I hope you are, please go and visit the CB Trust website. And every year we publish many scientific papers, but we also publish a statistical report on brain tumors in the United States, which I think the brain tumor community finds very valuable. And then, of course, we have partners all throughout the NCI, NIH, Health and Human Services, and externally, and we're very grateful for their collaborations. And I'm happy to take questions.
Jill, thank you so much. That was, I mean, it's an incredible amount of resources that you have and so many different data resources and options for tools and all sorts of things. That's really, really great. One of the questions I wanted to ask you was about the sharing of the data. Do you get the data from the grantees, the NCI grantees? Are they obliged to contribute their data as a condition of their funding? If anybody happen to have some data, could they contribute that too? Or are there any sort of rules of the road about their sharing?
No, it's a great question. So I think for any NIH grant, any NIH funded grant now, you're required to have a data management and sharing plan. And so the Cancer Research Data Commons is one place where you could share your data. It's cancer specific, right? So I don't think you're going to share your data through the Cancer Research Data Commons if it's not cancer data. But there are multiple generalist biorepositories that are available. Some are through the NIH, some are not. And so, yes, we do get a lot of inquiries through the data submission portal that I showed you from NCI funded investigators who are generating cancer data. I think if you're a cancer researcher and you have funding from somewhere else, it's non-NIH, we'd love to talk to you about what type of data you have and whether or not the Cancer Research Data Commons would be the right place for you to share it. Absolutely.
Yeah, in some projects that I've been involved with that are not cancer, but there's like if you've been funded by that institute to collect particular data types, like neuroimaging is what I work in, that you're obliged as a condition of your funding to then contribute that data back at some frequency back to the NIH so that it can be kind of disseminated, through their systems. This sounds like some of these resources do that for your cancer fundees, but that it's not impossible for somebody who's not funded by you to also possibly contribute their data. Would that be fair?
Yeah, I think we're as long as it's cancer focused, right?
Of
Because that's
course,
what
of
our
course.
mission is about. For sure we are open to talking with anybody.
Yeah, that's
The
great.
more data we have available, the more possibilities for making a difference, right? For individuals with cancer and their families.
Absolutely.
That's what
So
we're all about.
we had a couple of questions through
Great.
YouTube about what some of the most significant genetic mutations have been found in brain tumors through your TCGA. You covered several examples, but I'm curious if you have any other significant genetic mutations that you found through some of those resources.
Oh yeah, I mean, so the IDH mutation data and then the 1p and 19q chromosomal losses are really significant hallmark molecular features that I think we had known for a while that they were important in brain tumors, but they were not done at the scale where we could confirm them. I mean, so that's what TCGA really did for us. In addition, there are all these different defined molecular subgroups of the medulloblastomas. They have to do with some gene expression changes and also some fusion type changes, and that's how those different subgroups are defined. In addition, brain tumors have P53 mutations, just like every other cancer, does. P53, I think, if you read the statistics on it, it's one of the most important tumor suppressor genes in cancer, sort of period, right? Over 50% of all cancers have a mutation somewhere in that gene. In addition, there are some other oncogenes that are turned on, some in the PI3 kinase pathway. You don't typically see the RAS pathway activated in brain tumors. I would love to tell you that another really important molecular feature in malignant brain tumors is MGMT methylation. So MGMT is a DNA repair gene. And the chemotherapy that we give as part of standard of care for gliomas targets that gene. It's called temozolomide. It's very interesting, however, that gliomas do not have, when you look sort of at the burden of somatic mutations by different types of cancers, gliomas are sort of somewhere in the middle. They don't have as high of a burden as some other cancers do.
What's kind of interesting about your IDH mutations is that using those data, using some of the approaches you described, there's been an opportunity there to change the way in which diagnostic identification and the identification of treatment pathways has happened. I think that's like, it has to be one of the most exciting aspects of this, is that it was only through the accumulation of all that data and then performing these kind of data science based approaches that you're able to do that, and that now we're smarter, right, about how we deal with that, and then we can like kind of target people in the right places rather than some one size fits all kind of a thing. Is that fair to say? And are there other examples of that you've seen, kind of little success stories?
Yeah, I mean, it definitely was a game changer, right?
Yeah.
I mean, it completely changed the structure of the diagnosis requirements, right? So I'll give you a very concrete example. So oligodendroglioma is a very specific type of glioma, okay? And before all this TCGA work was done, I think that there was debate, on what features, what morphological and topological features needed to be present to diagnose an oligodendroglioma. Now they have to have an IDH mutation and they have to have 1P and or 19Q deletion, period.
Mm-hmm.
Right? So now it's a little less fuzzy, right? It's a little more clear. And it's really important to get a diagnosis right? Because everything that comes after the diagnosis, for cancer patients, everything comes after the diagnosis, how you help them, right? So we need to get the diagnosis right. It's really important. TCGA, I gave you a specific example in brain tumors because it's what I study, it's what I know best, and I was involved, I had the pleasure of being involved in those studies as an author and a disease expert participant. But if you look at almost any of the TCGA papers, the initial sort of what they call marker papers, there have been these types of discoveries and impact on diagnosis and or drug discovery and or outcomes prediction for almost every one of the cancers that's included in TCGA. So it was a game changer. And that's why it was so important to make sure that the data were available to everybody, right? Because these data sets are really important. They've led to a lot of discoveries. And a lot of people now leverage the TCGA data sets in some interesting ways, right? One would be as a hypothesis driving kind of data set. I have a question, I ask it of the data I see how many individuals has this cluster of molecular changes, um, because I think these specific pathways are important. The second is they take the data and they merge it together with data that they've generated to create a bigger data set, more robust data set for themselves. And the third is as a validation set, right? So they've generated their own data on a set of individuals. They've come up with a result, and then they need an ancillary data set to confirm. And so they're using the data set as validation. So it's really exciting.
I love this notion that it's a tool for discovery science, right? Is that so often, you've got a hypothesis, then you have to go collect your data, and it takes a long time, and you have to go through that whole process. But here, it really is kind of at your fingertips. And you described a whole bunch of different tools. You described the biomedical data fabric toolbox. You just described some of the intuitive tools. I think that just makes it more accessible, doesn't it?
Yeah, I mean, the Cancer Research Data Commons will be, a platform that we hope to be implementing some of these tools that are generated from the Biomedical Data Fabric Toolbox. So we're very excited about that. This is all about, at the end of the day, Jill Barnholtz-Sloan saying this, of course, data is an enabler. And I believe it is our responsibility, to make sure that we are making the data available in a way that anybody can use it. Doesn't matter who you are, what your training is, what your focus is. Like we need people with questions, with the drive to ask questions in order to make a difference for cancer. We just
What,
do.
yeah, what do you see as a opportunities for people who are data scientists now, who are looking for opportunities to develop, to innovate in this space? I mean, it sounds like there's lots going on. Is there still room for people with original ideas to come in and contribute?
Oh, I think there's a lot of room for people with original ideas, to come in. I would recommend to anybody who's in data science training right now, like you need to know all this AI stuff, specifically the large language models. You really need to understand how they work and how you might be able to leverage them. I think you need to understand, obviously most data scientists I know, program in multiple different languages, right? Which is important to be flexible in that way. And then, make sure whatever you're doing has some, I'm a biostatistician by training, so, has some statistical rigor to it as well, I think is really important. But I think at the end of the day, it's about just, I always encourage every person I've ever interacted with, whether they're a trainee or a junior faculty or even a senior faculty, right? Keep a notebook or a document on your computer. Just keep writing down your ideas, right? Like as you're reading papers and as you're hearing people giving talks, just keep a running list of all your ideas and then revisit that list and start to think about what data for a data scientist, right? What data is already out there that you could grab, and use to try to answer the questions that you have.
Absolutely. One of the things in our Biomedical Data Science Innovation Lab activity is we're bringing together people who are working in this case of spatial multi-omics and genetics, and probably lots of people who are working in cancer, and data scientists, and bringing them together. And data like these that you just walked us through are some of the grist for that mill. And what they will do through our 5-day activity is they'll pull together what we like to call, we actually have them kind of work on something we call a grantlet, which basically follows the same sort of ideas as, say, like an R21 type class NIH grant. and with the hopes of getting enough of the raw material that they could go back to their labs, work together remotely after they've met together, to basically flesh it out into an R21 and submit it. And I'm curious if there are opportunities through, maybe you don't know, but if there are opportunities through the NCI to apply for grants like that, that they could, basically do some work in spatial multi-omics with data science and apply for funding. I don't know if you know or not, but it'd be great if you did.
Yeah, I will say I really don't know. CBIT does not do anything with grants. We don't have granting authority through CBIT. We really are a collaborative center that we, work across the
Fair
NCI
enough.
agencies. So
Tell us a little bit more about CBIT in our final minutes here.
sure. So CBIT is the Center for Biomedical Informatics and IT. CBIT is where the NCI CIO sits, Jeff Schilling, and he runs all of the foundational IT and the scientific IT for the NCI. So we do quite a bit on data platforms and data availability with the NCI Clinical Center and also with the intramural research investigators. In addition, we have the Informatics and Data Science program, which has 3 branches. So the Clinical Translational Research Informatics branch, which I talked to you about some of their work, it's led by Umit Topologu. We have the Data Ecosystems branch, which is where the CRDC sits, and that's led by Tanya Davidson. And then in one of the last slides where I was talking about the whole slide imaging omics, that is through the Cancer Genomics and Bioinformatics branch led by Daoud Mirzamin. In addition, we have an Office of Business Operations. We do a lot of sort of business operations through all of our collaborations through the NCI. So we do a lot of stuff.
That's great. How many people are part of the your team?
In CBIT, we have about 80 federal employees, and then we also have a variety of contractors that we work with.
That's fantastic. I hope that they're open to possible collaborations too. There may be some people who are really interested in this space and interested in learning more about how they can help contribute to the development of some of these tools and interfaces and other things. So,
Yeah, I mean, I would be thrilled to hear from anybody. I'm pretty easy to find with the last name Barnholtz-Sloan if you Google me. And please feel free to email me, and I'd be happy to talk with you, connect you with other people.
that's fantastic. Well, on that note, Jill Barnholtz-Sloan, thank you so much for joining us. We really appreciate you having a chance to talk about this really amazing set of cancer based resources that I'm certain people are going to want to be diving into. It just sounds really rich and exciting for their discovery science and for really downloading some of that data and applying some of their data science methodologies to it. So thank you for sharing that with us, Jill.
My pleasure. Thanks so much for having me. Hope everyone has a good weekend.
You have a good weekend.